Hello, everyone, and welcome to today's live broadcast, Whole Genome Sequencing as a Valuable Clinical Tool for the Management of Cancer Patients, presented by Dr. David Smith, Professor and Consultant at Mayo Clinic. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom/center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing educational credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Smith. I will turn over the presentation to him now.
It's a pleasure to be here to talk about whole genome sequencing, which can be abbreviated WGS, and I'm going to talk about how I believe this is going to very quickly become a valuable clinical tool for the management of cancer patients. If we think about DNA sequencing, the real birth of DNA sequencing was the development of dideoxy sequencing that was developed by Sanger. And in this slide, this shows the 4 different sequencing reactions that are done in dideoxy sequencing to be able to figure out the determination of where all of the A's, all of the G's, all of the C's, and all of the T's are. And with this technology, with 4 separate reactions, one can discern the sequence of a small stretch of DNA. After the DNA sequencing reaction is done, this is run on a gel, and you can see the bands at the bottom of the gel, and by reading up the bands, and you can see at the very bottom, you can determine the sequence of that DNA fragment. The limitation of this technology is the ability to separate the different size DNA fragments, and with this technology and long sequencing gels, one could sequence DNA fragments in length up to 600 to 800 bases in length. However, if you want to sequence something much larger, like an entire genome, the next slide shows the actual strategy that individual DNA fragments that are overlapping are sequenced separately, and then one assembles them together by looking for overlaps. Now, there was some discussion a number of years ago when people were talking about doing this for something as complex as an entire genome, but it turns out that this approach, which is known as shotgun sequencing, turned out to be the viable approach for the sequencing of an entire genome, even one as complex as a human. Now, using this technology, realizing that one needs to do 4 lanes for each DNA fragment to be sequenced, it turns out that to sequence the human genome, which is actually 6 billion base pairs in size, because we have 3 billion base pairs of DNA from our mothers and 3 billion from our fathers, you actually, because you are doing this shotgun sequencing, you actually have to sequence more than 6 billion base pairs. It's usually around 50 to 60 billion base pairs. However, with this technology, this would have required 222 million lanes, which would be at least 20 to 30 million gels, and the cheapest thing I can think of as a graduate student, it would have taken 1,000 graduate students working for 10 years to do this, and this just is not a viable strategy. However, with the development of a fluorescent DNA sequencer that was developed by Applied Biosystems, which enables you to actually do all the sequencing reactions in a single lane and then place this into something, and this is shown here, the results of a fluorescent sequencer, you suddenly have the capability of ramping this up. Now, this was then put together, and instead of running gels, they started running capillaries, and then the evolution of this all the way to this machine, which was the Applied Biosystems 370, which in 1998 was the most advanced sequencer on the planet. You have an automated sequencer that is capable of doing 96 simultaneous sequencing reactions. With this, in a 3-hour runtime, the output on this machine was 75,000 base pairs. Now, a single machine is clearly not going to be enough to give you the power to do a genome, but by putting 330 of these machines in a single room, and this is a picture actually of a company that was actually doing this sequencing, Celera Genomics, and this is in Gaithersburg, Maryland. This is now a picture that is over 20 years old. You see a room with 330 of these machines, and this becomes the factory for DNA sequencing. This was a significant advance over any of the sequencing that was done previously, and you can also see that there are very few people in this very large room. This is very much an automated factory, and by running 330 machines simultaneously and continuously running them, you can generate sufficient sequence that in a relatively short period of time, it turned out to be about 3 months, that you could generate the necessary 60 billion base pairs of DNA sequence. Now, the strengths and weaknesses of the sequencing by Sanger, which is also known as CE or capillary electrophoresis, the strengths were that it was highly accurate, because Sanger sequencing was about 99.99% accurate. It turns out that they didn't just do the shotgun sequencing, but they leveraged other technologies, including genetic linkage, physical mapping, to develop the genome sequence. But the weaknesses were that it was extremely costly. This cost altogether, the development of the machines, the buying of the machines, about $3 billion. And it was still 3 months of sequencing and multiple months for the assembly of the resulting sequence. Now, in order to take this type of technology and bringing this sequence to the masses, it was necessary to figure ways to dramatically decrease the cost of genome sequencing. It raises the question of how much for a genome before it becomes a standard part of clinical practice. It's kind of a trick question, because if you're just saying you want to sequence everyone on the planet, you probably would think of a very cheap sequencing cost. But if you have a child which is sick and you're very much worried about it, I'm sure you're willing to spend considerably more. But in reality, you still have to figure out ways to bring the cost of genome sequencing down, not just 1 or 2 orders of magnitude, but almost 1 million to 10 million times. But in order to do that, there are 2 main problems for capillary CE sequencing. The first is that each individual fragment had to be cloned into E. coli, and the second one was that each individual sequencing reaction, which by itself doesn't seem like a lot of volume, 10 microliters, but that still uses a considerable amount of reagents. So how can we solve these 2 problems? Well, the first one is by figuring a way to replace cloning. The purpose of cloning was the amplification of an individual fragment that was not contaminated with other fragments. Now, the solution is to use some sort of PCR strategy, but you still need a strategy where you amplify individual fragments. If you take an entire genome and all the pieces and just do PCR, you're going to be amplifying all the pieces together. So you need a strategy where the fragments are isolated from each other so that you can make a sufficient number of copies of each individual fragment to get enough of a signal for sequencing. And then there were actually 2 different strategies that were developed. The first, the first developed was Emulsion PCR, but I listed bridge amplification as the first one because it's the more elegant strategy. But you have bridge amplification and Emulsion PCR as 2 different strategies whereby you don't need to clone the pieces in E. coli, whereby you can actually amplify enough individual fragments to represent an entire genome. The second thing is to figure a way to reduce that 10 microliter reaction of capillary electrophoresis. Now, how can one do this? Well, it turns out the very first strategy developed was the use of picotiter plates. These are plates where the individual well in the plate holds a picoliter type volume. So we're talking about not 10 microliters or 10 nanoliters, but 50 picoliters, which is significantly less. Now, these pico-titer plates were actually developed by taking a fiber optic cable from the telecommunication industry, slicing it like a salami, and then basically putting a plastic coating on the bottom, so you have a plate where inside the plate there is 1.5 million wells. Each well will hold 50 picoliters of volume. The second strategy, which is not to use those pico-titer plates at all, but was actually to put clusters of amplified DNA fragments on a slide surface. The most important thing was to use a strategy, which is a very powerful strategy that was used in the computer industry, which is why computers are so powerful today, and that's the fact of using massively parallel computing. Instead of having a single processor chip, you have millions of processor chips. And then the same strategy for sequencing is, instead of doing an individual sequencing reaction, or like on an ABI 370, 96 simultaneous sequencing reactions, is to do something where you could actually do hundreds of thousands to millions to ultimately billions of simultaneous sequencing reactions. The first generation were pico-titer plates, and those contained 1.5 million wells. Now, massively parallel sequencing thus sparks a revolution. You amplify individual fragments to produce enough template for sequencing. You sequence hundreds of thousands of these amplified fragments simultaneously. You utilize considerably less sequencing reagent per amplified fragment. And all of this together, the use of massively parallel sequencing, is known as next generation sequencing, or abbreviated NGS. Now, this next slide shows what is done for bridge amplification. For bridge amplification, the entire fragments for a genome are fragmented and to a very relatively small size, and then there are sequencing primers that are put on the ends of each fragment. You could call one color the pink one A and the blue one B. These DNA fragments are then flowed over a cell surface, and this surface actually has complementary oligonucleotides that are complementary to the A and B oligonucleotides that are placed on the end of each DNA fragment. One end of that DNA fragment will find its complement and will actually be able to stick to that, and now you have a DNA fragment that is tethered at one end, but at the other end there is still the other oligonucleotide, and that can actually find its complement on the slide surface. And when that happens, you actually have a DNA fragment that is now bent over, where it has an A on one side, a B on the other, and now you have a template and a single DNA fragment, which in the presence of polymerase could make a copy. If you then take that and denature it, you have two fragments, and those can also bend over, and by repeating this cycle about 30 times, you can actually do something where you're amplifying each individual fragment to, on this strategy, about 30,000 copies. But across the entire surface, you have a series of different amplified fragments that are now 30,000 copies where there was a single one, and each one is separate from every other one, so you have this capability of amplifying each individual fragment without any contamination from another fragment. The other strategy is known as Emulsion PCR. It is very similar. You take your DNA, and the DNA is again fragmented to small pieces, and you put the two oligonucleotides at the ends. So this is very similar, but instead of doing this on a slide surface, what you do is you actually do this in a water lipid emulsion. And in this emulsion, if you sonicate water and lipid together, you can actually produce these very tiny little water droplets in the lipid, and they are also picoliter in volume. Now, in this, you also have a very tiny bead. First generation of this was a 28 micron bead. On the surface of the bead are these complementary oligonucleotides. So each individual bead is very similar to the surface on the flow cell of the previous strategy. And in this bead, with a DNA fragment, you have the capability of amplifying an individual fragment to a very large number of copies. With a 28 micron bead and this type of strategy, you could actually make 1 million copies of each individual fragment. You do this in a very dilute solution so that most beads don't amplify any DNA, because you don't want the concentration of DNA to be so high that beads are amplifying more than a single DNA fragment. but it's very easy to purify those beads that have amplified DNA from those that do not. So after this amplification process, you break these emulsions, you purify the beads, and then you use basically a weight selection, like something like a sucrose gradient, to separate those beads that have amplified DNA fragments that those that don't. And yet these beads are so small that one can produce billions of these beads in a 1.5 mil microcentrifuge tube. Now, the very first next-generation DNA sequencer was developed by a company called 454 that comes out of Connecticut. Their first machine was called the GS20. It was called GS20 because it was a genome sequencer, and the 20 referred to the fact that the output from this machine when it first came out was 20 megabases. Now, this is a significant advance because I mentioned earlier that the ABI 370, which was the most advanced DNA sequencer in 1998, had the capability of doing 75,000 base pairs per sequence run, whereas this machine, when it first started, had an output of 20 megabases. So this is significant. It's basically a 500-fold increase in output. This machine is coupled with, which is shown on the next slide, the emulsion PCR. So the individual DNA fragments are amplified in these separate emulsions. Those are purified, and then these individual beads with amplified DNA are basically placed onto this pico titer plate that has some 1.5 million wells. Each individual fragment is then placed in these wells, and in addition to these amplified 28 micron beads are all the necessary reagents, the DNA NTPs, the polymerase, etc., for the sequencing reaction. It's placed into the pico titer plate, and it's placed into the machine, and it's set up in such a way that you can then flow one at a time individual oligonucleotides. So individual nucleotides, excuse me. So first you place an A and you see which one of these beads is incorporating an A as the first base. The enzymes that are set up for this are set up so that when a base is incorporated, there will be a flash of light. This is known as pyrosequencing. So you flood the entire pico titer plate with an A, and you ask which of the wells has given a flash of light. Then a C, a G, and a T, and that's the interpretation of the first base. If one then utilizes this, on the right, one sees the output from one of these single beads, but you have to realize that across the entire pico titer plate, there are at least 500,000 of these beads with an individual DNA fragment. And on this, you can actually see the strength, but also the weakness of this technology, because the nucleotides themselves are unblocked, which means that if you have a string of A's, for example, that entire string of A's is going to be incorporating T's. So if there's only a single base that's incorporated, you see a peak at a certain height. As you go across that top slide, you see suddenly a peak where there is multiple nucleotides being added, and one can tell the difference between a single base being added, 2 bases, and 3 bases. But if one has a long homopolymer stretch, a long enough stretch, it's very difficult to tell the difference, for example, between 10 and 11 A's. But one of the nice things about this technology is that it gives a strong signal because there is 1 million amplified DNA fragments on each bead, and one can get a long sequence read. When this is showing 191 base pair read, but with this technology, it was possible to get reads that were very similar in length to the length of reads that you were getting with Sanger sequencing. The strengths and weaknesses of this were there were long reads. When they started, there was 400 base pairs. There were rapid improvements to this technology. But the weakness was the emulsion PCR is a very messy technique that requires this lipid-water interface. The fact is that you have homopolymers because the nucleotides are not blocked. And the biggest problem was there was limited headroom for further increases in sequencing output. Now, when this machine started, it was called the GS20. Very quickly, it was capable of 40 megabases of sequence, and then 100 megabases. And actually, through the life of this instrument, it increased its output, but what happened was, very quickly, they reached a point where you couldn't get any more sequences on this instrument. So by the time this thing had reached its penultimate instrument, we had something that was known as the GS Flex Titanium. The wells themselves were coated with titanium, but it was capable now of 500 megabases of sequence output per run at a cost of $6,000. But it was not possible to get further output. However, Whereas the very first genome cost almost $3 billion to sequence. In 2007, as a promotional thing, they sequenced the genome of Jim Watson of Watson Crick fame. And on this platform, utilizing a 500 megabase output per run, the cost for sequencing Watson's genome was just $1 million. So we've already seen a dramatic improvement of basically $3 billion for a genome down to $1 million, but clearly our target is going to be a much less expensive genome. We could argue about what is the cost of a genome, at which point it becomes very useful for routine clinical practice, but it is clear that this improvement, which is basically a 3,000-fold improvement from Sanger sequencing, we're still going to have to figure out a strategy to improve this multiple orders of magnitude better. Well, it turns out Illumina, and this is the very first Illumina machine that was produced. This was produced in 2007. This was known as the GA for the Genome Analyzer. This platform utilized the different way of amplifying DNA, and this was amplifying DNA using bridge amplification. So using bridge amplification, one can amplify individual DNA fragments. On the left of the slide is the actual flow cell that is used in the very first generation Illumina machine. It's actually still used in the current generation Illumina machines, which is basically a flow cell that has 8 separate lanes. This enables you to put separate reactions in each lane if you wanted to. You could put the same reaction in multiple lanes if you wanted the output to be greater. And then this flow cell can be placed in a machine. Now, the nice thing about the Illumina platform, in addition to using a much better way of amplifying DNA in bridge amplification, is the fact that they are using individual labeled nucleotides that are a different fluorescent color. So the G's are one color, the C's, the T's, and the A's are different colors. The color is a fluor that is attached to each individual nucleotide, but it is nicely added at a position where when a nucleotide is added, the fluor is blocking the addition of the next base. So this blocking enables you to put one base at a time. So you actually will add, you will flow all 4 together simultaneously. You don't have to do one at a time across the flow cell. And then after that is done, you wash everything that is not incorporated away, and a camera will take a picture. The next step is to remove the fluorescent blocking group. By doing that, chemically removing that, the thing is now able to accept the next base, and one does that, takes a picture of that, and you can see on the right-hand side, the top one is the first base incorporated, the second one is the second base. You continue that process, you wash everything away, you take off the block fluorescence, and you do the third base, the fourth base, etc. And you can see, for example, the sequence of the little circle that is on the left is GCTGA, and that is the sequence of that base, but simultaneously you're doing the sequence of all the other amplified ones at the same time, hence the massively parallel sequencing. Now, the advances on this platform, the original machine was a significant advance because it was capable of a gigabase of sequence. And remember that the 454, even its penultimate machine, was only capable of 500 megabases. But to do this, we had 48 million clusters, but the size of the sequence that was generated was only 31 base pairs, and these were relatively short. However, over the next 3 years, they increased the number of clusters that could be read, and the read length of individual fragments that was sequenced was up to 100 base pairs, and this resulted in a dramatic increase in the number of gigabases that could be sequenced and at a higher accuracy. However, 4 years after the very first machine came out, an improvement to this came out, and this is shown here, and this is the HiSeq 2000. This machine had a number of improvements from the previous machine. Instead of doing a single flow cell, the realization was that while you're doing the chemistry on one flow cell, it would be a good time to actually take pictures of the other flow cell. So this machine actually ran 2 flow cells at a time. In addition, the clusters that were produced on the flow cell turned out to be not just on the top of the slide, but on the bottom of the slide. So the HiSeq 2000 had the ability to actually read both the top slide and the bottom of the slide. So this increased the output dramatically. These machines had even greater output, but as shown on the slide here, at the bottom right is after a while, you start to produce so many clusters on the slide surface that it becomes very difficult to tell one cluster from another. How does one increase this so that one can get even greater output? And the answer to that question was to, instead of just having a flat slide surface on the flow cell, is to have a pattern flow cell technology that is shown on the bottom left, where each individual fragment is not amplified on a flat surface, but in an individual well. And then when one does sequencing with this, and if you see the bottom and the middle, you can see that it's very easy to discern one sequence site from another, and this results in the ability to get even higher densities. Unfortunately, in the same way that the very first generation machine had a very short life, the HiSeqs themselves had a short life with the replacement of these pattern flow cells, and each of these machines was costing $700,000, but 2 to 3 years later was obsolete because of these advances in technology. The next slide shows the current version of the Illumina machine, which is known as the NovaSeq. The NovaSeq is using the pattern flow cells again. There's actually 2 versions of this machine that is shown on the next slide. The NovaSeq 5000, which runs a single slide, and the NovaSeq 6000, which runs 2. And one of the nice things about this system was the output was even greater. Remember, the first generation Illumina machine was capable of a gigabase of sequencing, but on the NovaSeq, with the smallest flow cell, you're capable of producing 500 gigabases, or half a terabase. Recently, the 3 terabase flow cell has come out. This means that one can do 6 terabases, or 6,000 gigabases of sequence per sequence run. So the output on this machine is absolutely incredible. Now, with this type of technology, the capability of doing 6 terabases of sequence every time you run this machine, you have the capability of doing a lot of different types of sequencing. One type of sequencing, which really is the major subject of what I'm talking about today, is whole genome sequencing, or WGS. To do WGS, you need to do about 100 gigabases of sequencing per normal genome. However, there are many reasons why you may not want to do that much sequencing, and one possible solution is just to sequence defined portions of the genome that you want to look at. And in a clinical setting, that might be a group of genes that are involved in the disease that you're actually studying. If you want to sequence less than a whole genome, but don't want to sequence just a very small portion, one can do whole exome sequencing, which is known as WES. And this is the sequencing of the 200,000 exons that comprises the coding portion of the genome. Another thing that can be done with this technology is transcriptome sequencing. This is actually the sequencing of what is actively being transcribed in cells, and it's basically taking the RNA from cells and converting it into DNA, and one uses a cDNA approach towards this, and then one can figure out what is being actively transcribed in the cell. One can also not just sequence the DNA, but can sequence the DNA that has been epigenetically modified by methylation, and then one can determine portions of the genome that are more susceptible to environmental influences. So you have, with the capability of a NovaSeq machine, the ability to do a variety of different types of sequencing, and then one has to evaluate these to say which one of these is best for which specific use. Well, whole genome sequencing, as I said, requires one to sequence at least 100 gigabases if one is sequencing a cancer genome. And if that cancer genome has heterogeneity, and this is seen in a number of cancers, most notably in breast cancer, you might need to sequence more than 100 gigabases. If you're looking at a population of cells that is only 10% of the total, and you want to see them and sequence them fully, you're going to probably need to sequence up to a terabase of sequence. But once you've generated the sequence on these machines, you have millions of fragments and their DNA sequence, but that still needs to be assembled together. One must look for overlaps between these fragments. The other part, which is a very critical component, is to analyze the assembled genome, and one has to ask the question, what are you looking for? What types of alterations? Are you looking for alterations of individual nucleotides? Are you looking for alterations that comprise basically changes in the DNA, like amplification? Are you looking for translocations? All of these things add complexity to the overall problem. Now, for defined portions of the genome, again, one could ask the question, why sequence and assemble all 6 gigabases of your genome? What about examining specific regions or genes for specific tests? And again, it could be a small panel of genes, or it could be the entire exome, which is, again, 200,000 exons, but it's only 38 megabases. Well, the first question is, how does one enrich for whatever defined portion of the genome you want? And on the next slide, one sees the strategy that's actually done by Agilent, which is their Sure Select, but they're very similar strategies from other companies. And what one does is one produces, and this is shown on the top right, baits. And these baits are actually sequences that you want to capture. They're biotinylated, and then the DNA from the entire genome is fragmented into small pieces. There are PCR primers, which are also sequencing primers, that are put on the end of each of these fragments. One uses the baits to fish for the sequences that you want. When you actually then put them together, you have pieces with the biotinylated baits, and this is the perfect thing to attract with a magnetic bead that's coated with streptavidin. The streptavidin will bind the biotinylated pieces, and then you can pull those pieces out with a magnet, and then the oligonucleotides at the end of the fragments can be used to PCR what you have captured. And the nice thing about this is whatever you want to do. If you want to capture the exons of 10 genes, you can do this. If you want to capture the exons of the entire exome, all 200,000, that can also be done in a very small tube. Now, how are the baits made? They're actually made using Hewlett-Packard-based printing, because Agilent is actually a company that comes from Hewlett-Packard. And one of the nice things about color printing is color printing uses 4 different colors. Well, the same thing that comes in groups of 4 are the individual nucleotides. So instead of making a color picture as you would with a printer, you actually use that same technology, but you put the 4 bases in that substitute them for the colors, and then you can synthesize on a slide surface millions of different long oligonucleotides, like you would sort of print a color picture. And then once that's done, you can actually remove all those oligonucleotides from the slide surface, because usually they're connected by a sulfhydryl bond. And then you have them in a tube, and you suddenly have the baits for the sequences that you would like to capture. Now, whole exome sequencing is basically baits for all 200,000 exons, which is, again, 38.5 megabases. The nice thing about this is on a NovaSeq machine, a small fraction of a lane is sufficient to sequence the entire exome of an individual or of a cancer, or whatever you want to look at. And still, you're going to read every single nucleotide that you've captured at greater than 200-fold depth. So the advantage of this is that you can do greater depth as you're sequencing it, and it requires so much less sequencing to capture an entire exome. If one uses a NovaSeq machine, which is capable of producing 6 terabases of sequence, that is sufficient sequencing to sequence 60 individuals for whole genome sequencing, but it's actually useful. You can do hundreds or multiple hundreds of exomes each time you run the machine. So whole exome sequencing is a much cheaper way to characterize a portion of the genome. Transcriptome sequencing is useful because whole genome sequencing, or defined region sequencing, or exome sequencing only tells you what's in the DNA and does not reveal what is actively being transcribed in a cell. So transcriptome sequencing analyzes what's transcribed. However, when one isolates RNA, 95% of that RNA is ribosomal, which you're not interested in. So one needs to figure a way to get rid of the ribosomal RNA, and that can be removed either by capturing the transcribed sequences, which, when they're coding sequences, are polyadenylated, or by using baits for the ribosomal RNA to remove them. And then once that's removed, you can sequence the rest. Now, what can you detect with RNA sequence? An RNA sequencing known as RNA-seq, you can see transcript abundance, but with a digital output. And this is so much better than microarrays, because you get a digital output, and it's possible to see transcripts that are even relatively rare. With microarrays, it's very difficult to see the bottom 50% of expressed transcripts. With RNA-seq, one can see almost everything that is actively transcribed. One can see transcript isoforms. One can see fused transcripts, which could be both isoforms and fused transcripts could be very important in a cancer context. And since you're actually sequencing each nucleotide, you're actually able to see mutations in the most abundantly expressed transcripts. And it totally depends on how much sequencing you do as to which transcripts you can see. With a low-pass sequencing, you can only see the sequence of the most abundantly expressed transcripts. With high sequencing, one can see the sequence of most of the transcripts. So this is a powerful technology. Methylation sequencing, and there's multiple different types of epigenetic modifications of the DNA, but the one we focus on the most is 5-methylcytosine. And this is the methylation of cytosine, which is, as I said, is indicative of environmental exposure. Well, an example just out of my own life, most older Americans become lactose intolerant because as they get older, the lactase gene becomes methylated. And this is why I can't eat ice cream or milk or any of those things, because my lactase gene has become methylated. Again, this is indicative of aging or environmental exposure, but one can look at methylation of a genome with this type of technology. Now, each of these has strengths and weaknesses. Whole genome sequencing has a number of strengths, but also some weaknesses. Again, with the NovaSeq and pattern flow cells, the cost to generate 100 gigabases of sequence is only $350, but this is just the cost for sequencing. How much does it cost to assemble the genome and interpret it? What about alterations outside of the coding sequence? Because only 2% of the genome actually codes for protein, but in a cancer cell, there will be alterations throughout the genome. What is the clinical significance of those alterations? Even within alterations with the coding sequence of the genome, which of those alterations are actionable? And this is a very small fraction. The other issue is that even after you've generated the sequence, even after you've assembled the sequence together and analyze it, how much does it cost to store that data? And thus, generally, this is not ready for primetime clinical practice in most settings. Now, the strengths and weaknesses of exome sequencing, its strengths is it's cheaper than whole genome sequencing. One generates much greater sequence depth for most bases. It's considerably easier bioinformatically because you have much less to analyze, and of course, there's much less data to store. However, because you're using a capture technique, which is not capturing every single nucleotide at the same level, you do not get 100% of the target bases. Sometimes there are bases that are not gotten at sufficient depth to be able to discern what their sequence is. In addition, one generates no information outside of the exome or whatever was targeted, and if you're trying to figure out anything other than the changes in the nucleotides, for example, copy number variation, it's difficult to take exome sequencing and determine what's going on in terms of changes in copy number across a genome. The strengths and weaknesses of RNA sequencing or RNA-seq is that it complements all the other technology. It's much easier bioinformatically, especially for message abundance, and of course, there's even less data to store. However, by itself, it does not provide information about genomic changes outside of the exome. Strengths and weaknesses of methylation sequencing is, even though the strength is it's a better indicator of environmental exposure, and it's very complementary to other techniques, especially RNA-seq, but in order to generate full methylation sequencing, this requires you to take the entire genome, bisulfite modify it, and one sequences the unmodified genome and then the modified genome, and one looks for the changes, and those changes are where there is a 5-methylcytosine. But the classical model of how methylation, specifically methylation of CpG islands, controls gene expression is being challenged, and actually many people are starting to believe that it could be plain wrong. So we don't really know which methylations across the genome are important for changing gene expression. Hence, a lot of methylation sequencing is going to generate data that we can't make any sense out of. Now, what is the cost of next-generation sequencing? And this is for all types of next-generation sequencing. The first cost is the generation of the library. And this totally depends on the type of library you're making. Now, one of the things is that one could figure a way to reduce this cost by making the reaction volumes even smaller. Instead of these 10 microliter sequencing reactions, could one reduce the reaction volumes to much less? Well, there's companies like GenCell or Advanced Liquid Logic that have the capability of producing libraries, but not in 10 microliter, but 10 nanoliter volumes, which means that one uses less reagents. There's the actual cost of the sequencing itself, and this totally depends on what you're sequencing and how deep you want to go. But obviously, if sequence output increases, the overall cost of sequencing decreases. The biggest bottleneck of this entire strategy is analysis, and it's a bottleneck of the entire technology, and it totally depends, again, on what you're sequencing. There's less analysis if one is doing RNA-seq, less analysis if one's doing a small gene panel. There's much more analysis if one is doing an entire genome for whole genome sequencing, or for the methylome if one's doing the entire methylome. And of course, another important part of a cost is the storage of the data. And again, which technology you use determines how much that's going to cost. If you're just doing RNA-seq, there's much less data. If you're doing whole genome sequencing, there's much more data. Now, What are the clinical uses of whole genome sequencing today? The very first use of it is in the NICU units, or the neonatal intensive care units, because a child is born, is failing to thrive, parents are obviously upset and hysterical, and they want to know what is wrong with their child, and in a very quick setting. Well, it turns out this was pioneered by Stephen Kingsmore and his colleagues, and now they have developed strategies where they've taken whole genome sequencing and are capable of doing this, I say 26 hours here, but I've recently read that this is down to 23 hours, and as a result of this, he's actually in the Guinness Book of World Records for the fastest turnaround time for genome sequencing, and this is important because you can basically take some DNA from a newborn, generate the whole genome sequencing, analyze it, and have the data and the analysis ready 24 hours later when the doctors are rotating to each of the patients to see what is wrong with that patient. And this is really transforming what's happening in NICU units. If you don't want to do whole genome sequencing, a cheaper strategy might be whole exome sequencing. But again, this is used specifically in the neonatal intensive care unit setting. And the whole genome sequencing is not yet being routinely used at all for cancer care. Now, clinical uses of whole exome sequencing. WashU in St. Louis has been using whole exome sequencing to pull down all the exons and sequence them. And even though they may not be interested in all 20,000 genes, they can focus on the genes of interest. For example, if you have a patient with a cardiovascular problem, you could focus just on the cardiovascular genes. But the nice thing about this strategy and technology is that one can develop databases of alterations in disease, and you could query regions that might turn out to be significant that you don't know about when you start. And it's not much more expensive than just focusing on small gene panels. So this is why this is a powerful technology when you don't want to do whole genome sequencing, but you want to still be comprehensive in looking at the coding portion of the genome. The next generation cancer panels are really the strategy that is used most in the cancer setting. For example, Foundation Medicine has a 315 gene panel. Unfortunately, they don't really make money, so they're actually doing this, but they're not generating enough money per test to pay for it. But what they are doing is they're developing databases of alterations in cancers, and this could be very powerful. This is sort of the Myriad model that Myriad used for BRCA1 and BRCA2. Memorial SmithKline and Hopkins are developing 400+ gene panels for cancer, and it's the same game plan as Foundation Medicine. They're not really making money on each test, but they're developing databases. Mayo Clinic, which is much more concerned about making money on tests and being fiscally responsible, has a 50 gene cancer panel, but they're now working to develop a 400 gene panel. Many places in Europe are also developing larger gene cancer panels, but of course for all of these, strong bioinformatics to support this is extremely key. Now, what about NGS for clinical cancer care? Currently, small gene panels are the sequencing strategy of choice in cancer care. Less DNA to sequence, much simpler bioinformatics, and it helps to target important genes and helps to identify actionable alterations. But one still has to realize, even with a 300 gene panel, the alterations that one detects, the vast majority of alterations are not clinically actionable. There is no drug currently available, even if a gene is mutated. Now, what are the problems with small gene panels? This only provides information about the genes that you target. No information about copy number alterations, no information about gene fusions, and the total cost includes the capture, which is not cheap. It actually turns out that to capture a small number of genes or to capture an exome is about similar in cost, and it still is a significant cost. And there's no information about alien DNA, DNA that is present there, such as viruses, unless that's actually put and targeted in the baits that you utilize. Now, whole genome sequencing for cancer care, a key strength is it provides complete information about a cancer. It identifies most of the single nucleotide polymorphisms, or SNPs. It identifies the copy number variants, the translocations, etc. Whatever DNA is present in what you're studying will be sequenced, so it identifies foreign DNA such as viruses, and it's a much simpler library prep with no need for enrichment. So actually the cheapest prep for any of the NGS is for whole genome sequencing. One simply takes all the DNA fragments, puts on the suitable sequencing primers, and basically takes that and sequence it. So it's actually cheaper to produce the library than some of the other techniques. Where it becomes more expensive is one needs to sequence more DNA to cover the entire genome. So what will it take for whole genome sequencing to become the clinical test for cancer? Well, the first thing is the total cost for whole genome sequencing must be less. For many years, people were talking about the $1,000 genome, but I'm actually a big fan of a $100 genome. And when I mean $100 genome, I mean total cost all in, so that everything, library preparation, sequencing, analysis, all together must be $100. Because at that point, small gene panels or whole exome sequencing, just the capture itself costs a couple, $100 to $200 for the panels or whole exome sequencing. Why do them when you can do whole genome and have comprehensive genome information for a fraction of the cost? Quicker turnaround time, but it turns out that's already been solved by Stephen Kingsmore and his work in the NICU units. But that raises the question, when will the cost of whole genome sequencing be $100 or less? One of the things I did not mention is that in the sequencing world, there is a monopoly that is now controlled by the company Illumina. Some 90 to 95% of the sequencing that's done on this planet is done on the Illumina platform. And without significant contribution, without significant competition, there is no motivation for a company like Illumina to generate a genome for a fraction of that cost. So the question is there some competition for Illumina? Well, it turns out that there is. BGI stands for the Beijing Genome Institute. Used to be in Beijing, but is now in Shenzhen, which is sort of the free zone in China. And basically, BGI has very ambitious plans. They have plans to do 1 million newborn Chinese people, known as their million newborn cohorts. They're doing 1 million plant and animal genomes. They want to do millions, multiple millions of microorganisms. And the CNGB, which is the China National Gene Bank, is an incredible facility that I've had the honor of being at. And they're storing 20 million samples. They're doing 1,000 plus sequencing machines. They have 8 petabytes of data that's generated each day. And this is a very big sequencing place. And they have a sequencing platform that actually could be competition, which is shown here. This was a strategy developed by Rade Dramanjic for the company Complete Genomics. They still needed a third strategy for amplifying individual DNA fragments, and the strategy that they use is called nanoballs. You take small little DNA fragments that are then circularized, and in the presence of a 5' polymerase, you can take those little circles and amplify them with a rolling circle amplification so that each individual fragment can be amplified to 300 copies. These amplified DNAs then curl up into these tiny little balls that are known as a nanoball, and you can see in a small little tube you can have 100 billion little nanoballs. Each individual nanoball can be placed on a spot, and on this type of sequencing, you have 4 billion spots per slide, with, in this case, 600 nanometer separation between the spots. And then these are taken, and DNA sequencing can be done on this. Initially, BGI would do all their sequencing in-house, but very recently, they've developed a machine known as the BGI Seq 500, which is a DNA nanoball-based efficient benchtop sequencer, and it is been approved by the China FDA. It uses stepwise sequencing by polymerase. The output on this machine, first generation, was about 300 gigabases, and there were over 400 BGI Seq 500s that have been deployed. Now there's a manufacturing component of this, and they're producing the MGI Seq 500, which is just the manufactured BGI Seq. And these machines are starting to be sent to various places. They're not yet currently available in the United States. There may be some issues of patentability in terms of how the sequencing is done, but this machine has the capability of producing types of sequence output that begins to be comparable to what's done on Illumina. Now, it turns out BGI is now willing to do whole genome sequencing for a total cost of $600. But by the end of this year, that is going to be reduced to $200. And they are targeting the $100 genome total all-in cost by the end of 2019. And this is significant. Competition is thus good, because as BGI ramps towards providing a $100 all-in human genome, Illumina will have no choice but to try to compete. And thus, we've seen a reduction in whole genome sequencing costs from $3 billion to $600 down to $100 in about 19 years. And once we have the $100 genome, one can ask the question, well, when was that going to be? Well, all we need is a 10-fold improvement to the cost of a genome is just $10. And maybe that'll be the next target. So whole genome sequencing for cancer, easy library prep, ultimately less expensive than any other next-generation sequencing approach. It provides complete genome information, including SNPs, CNVs, translocations, and foreign DNA. And whole genome sequencing data in cancer, well, one could take the data that's one's generating with these $100 genomes, and the very first thing one can do is molecularly characterize each cancer. As we're starting to see out of projects like the TCGA project, by sequencing sufficient numbers of cancers, one can use molecular classifications that are already turning out to be more powerful than pathology characterization to get molecular subgroups of each cancer. This is first seen in breast cancer and also in lung cancer, and one can see that different types of molecular characterizations of different cancers will have different outcomes and also different treatment strategies. Now, the different subtypes, again, will have different treatment strategies, and this can still be used to identify actionable mutations. The only difference is that such a small proportion of the total number of alterations that one sees in a genome are going to be actionable. Even a small percentage of a 400 gene panel are actionable, an even much smaller percentage out of an entire genome. Now, what is the problems with whole genome sequencing for cancer? Again, most alterations detected will be outside the coding portion of the genome. What do they even mean? However, there was a very interesting recent paper that came out of the Rule Shanayan lab where they're showing a long non-coding RNA which is involved in prostate cancer that itself might be a target. So as more and more sequencing is done, as more and more work is done on the alterations that one sees in cancer, we're going to start to find alterations outside of the coding portion of the genome that themselves could be potentially actionable. But even a small proportion of the alterations in the coding portion of the genome are currently actionable, which means that we need many more drugs for mutating genes to really be useful. We're also going to have much more information about how multiple drugs that are targeting different alterations work together, how toxic they are, etc. And there's a huge amount of data, which is way too expensive to store the full sequence. So we're going to need some solutions of maybe just storing the important alterations or figuring out different ways where we can bring down the cost of data storage so that one doesn't have a $100 genome that costs $10,000 to store in terms of data. However, whole genome sequencing provides information on cancer-specific mutations, and cancer-specific mutations are valuable markers for monitoring cancers. And this can be used in two different ways. One is as a potential diagnostic tool for cancer screening, and this is being employed by the company Grail that is trying to develop tests where they can screen populations of individuals for known alterations found in cancer. But more importantly, until that has been developed, one can still use this to detect the presence of cancer cells and potentially the amount of cancer present in an individual undergoing clinical treatment. And this brings up the entire concept of the liquid biopsy. Since metastatic solid tumors, and actually blood tumors as well, have those cells in the blood, the circulating tumor DNA that is present, it can be detected in the blood. The half-life of this DNA is very short, which means that if you figure a way to eradicate or decrease the number of cancer cells where the cancer is, that the circulating DNA that came from that cancer with a short half-life is going to decrease very rapidly. Since the concentration of circulating free tumor DNA may be very low and is only a fraction as compared to normal DNA, how can one detect such rare DNAs? Well, it turns out that can be done with a number of techniques, but one powerful one is known as digital droplet PCR, or ddPCR. This can be used to identify rare sequence variants, such as rare molecules that have mutations, and the way this is done is very similar to the droplets that we use for emulsion PCR. You produce these individual droplets and you put very dilute DNA in the droplets, and there are two platforms that were available. One was this Bio-Rad QX200 droplet digital PCR system, which produced some 50,000 droplets. The alternative was a Raindance system, which produced up to 10 million droplets. Well, it turns out Bio-Rad bought Raindance so that they can control this entire market, and you basically have technologies where you can produce large numbers of individual droplets. You dilute the DNA that's isolated from the blood into the droplets, you amplify them, and as shown here on the next slide, you take their DNA, you amplify them, and you are looking for specific alterations that are present in that DNA. With these types of machines, you can look for as many as 5 or 6 independent alterations. You do the PCR, then you read this thing and you say, how many of these droplets contain this altered nucleotide? So this is a nucleotide by nucleotide examination. And one can say, if you see one droplet that has an altered DNA in the presence of several thousand that are normal, that is the concentration of the mutant molecule to the total. And then one could use this to monitor an individual who's going through treatment. So to use ddPCR to monitor cancers, you identify your tumor-specific markers. It could be a mutation in the tumor or a novel junction. The metastatic tumors, there will be circulating tumor DNA present in the blood, but it'll be a low concentration relative to normal. But one can isolate this, optimize for circulating DNA, and then one could look for whether there are mutant molecules. And this also produces a digital output because you can tell the ratio of the mutant droplets to the normal droplets, and that tells you the concentration, and then one can monitor by going through therapy. So for example, you identify your cancer-specific alterations for whole genome sequencing data. You construct PCR primers for these alterations that are patient-specific, and then one's ddPCR on the blood samples to monitor the patients post-surgery as well as post-other treatments. And if, for example, the surgery has been effective, one has eradicated all the cancer very quickly, there'll be no circulating DNA from the tumor, and one could say that that patient has no more circulating free DNA. Perhaps the surgery's been effective. If you're using this to monitor other things like chemotherapy or radiotherapy, one can still do this to see in real time how patients are responding to these therapies, and one gets a very quick feedback, and one can tell if a therapy is working or not. If a therapy is not working, why treat patients with toxic compounds? So whole genome sequencing has the potential to completely change how we treat cancer patients. When the total cost of whole genome sequencing is $100, it will become the technique of choice for characterizing cancers. It's cheaper and easier than gene panels or even whole exome sequencing. It's a comprehensive way of examining genome-wide alterations. One can focus on those alterations that are actionable and simultaneously use the data generated to molecularly classify each cancer. One can thus use the liquid biopsy to follow cancer treatments in real time. So this is the end of my presentation. Basically, these technologies are going to completely transform how we treat cancer patients. It is my firm belief that when the cost of a genome is $100, we're going to rapidly see the adoption of this as a methodology for cancer patients. Actually, we're going to start to see this as the methodology that's used in the normal population as well. And in the next couple of years, we're going to see a transformation in how we treat cancer patients. We're going to start to treat with a very personalized approach individual cancer patients to understand what's altered in their individual cancer, to molecularly classify them, then to find the right drugs for treating that cancer and using the liquid biopsy to monitor. So this revolution is exciting. It's exciting to be a part of it. And I advise anyone in the population is thinking of getting cancer to just wait a few years, because our ability to treat cancer is going to be so much better 5 years from now than it is today. And I thank you for your attention.
Thank you, Dr. Smith, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question is, how does next-generation sequencing compare to Sanger sequencing with respect to sequence accuracy?
This is an important question. When next-generation sequencing first started, it was not as accurate as Sanger sequencing. So people thought of Sanger sequencing as the gold standard and next-generation sequencing as being less accurate. However, the advances in next-generation sequencing have gotten to the point now where, because of the sequence output that is possible, and by sequencing each individual nucleotide a sufficient number of times, one gets a similar type of accuracy as Sanger sequencing. And as a result, my prediction is that Sanger sequencing is going to, over the next couple of years, be replaced almost completely by next-generation sequencing.
All right, thank you. Second question, Will next-generation sequencing replace Sanger sequencing, and how soon?
Well, the Sanger sequencing for molecular tests has over the past 10 years really become extremely popular. But as people start to use next generation sequencing and start to do gene panels, we're starting to see at the Mayo Clinic, it's starting to replace Sanger sequencing. So my guess is over the next couple of years, we're going to see more and more next generation sequencing and less Sanger sequencing, unless you're just interpreting and looking at a single gene, for example. If you're doing a cystic fibrosis test, there really is no reason to do next generation sequencing. But over the next couple of years, NGS is going to replace Sanger sequencing.
Why are small gene panels the current next generation sequencing technique of choice right now for cancer patients?
There are a number of reasons for this. The major reason is cost, because the total cost of whole genome sequencing is still too great when one factors in everything, including data analysis and storage. The second thing is that even a small fraction of the small gene panel have alterations which are actionable, and as a result, it becomes more expensive to do whole genomes and when you're only looking at a very small fraction of what is altered. And even a company like Foundation Medicine that's doing their 315 gene panel aren't getting their money back. So I think why we're seeing the small gene panels are used now, because it's just easier to handle, less data, less storage, less bioinformatics. But as whole genome sequencing becomes cheap enough and we develop some strategies to solve some of the problems, we're going to see a transition in the same way that today we're seeing Sanger sequencing going to next generation sequencing. We're going to see small gene panels go towards whole genome sequencing.
Okay, and we have time for one more question. What will it take for whole genome sequencing to become the clinical test for cancer?
As I discussed in my talk, I think when the capability is that you can do an entire genome for $100, I think we're going to have very little reason to do small gene panels or exomes. There still are significant problems, however, including data storage, including better ways to very quickly go to the alterations that you want to. But again, when a genome costs $100, I think we're going to see a transition. And it's my prediction that if we come back in 5 years' time, there's going to be an awful lot of whole genome sequencing that's going to be used in the cancer patient setting.
All right, I would like to once again thank Dr. Smith for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.